Okay. Karen, you are there? You didn't do the well in this one here. Maybe you didn't also read there. You are there in the lesson.
Yes, I'm here.
Okay. Well, we want you guys to pass, do well. But the earlier the better. Now, look at this. So which graph shows you the effect of enzymes of temperature on activity of enzymes? Now, if you are reading, you know that high temperature denatures what? You can't have a graph like this. Because at high temperature, above 50 degrees, the enzyme will be destroyed. So there won't be any activity. So this is the
Yeah.
correct one there. Because high temperature denatures enzyme. Are we together? It completely destroys the enzyme, so there won't be any activity. Then what we said is that human digestive enzymes are denatured at 50 degrees. But you might
Yeah.
find that for animals in the desert, it might be higher temperatures than this. Clear? But the idea, the shape of the graph will still be the same. Because at a certain temperature, the enzyme
Yeah,
is
I want to talk to you.
completely destroyed. That making sense there, Norma? Then
Yes,
these
sir.
guys are saying, but this one is missing. Yes, it's true, it's missing, but we can get the answer straight away. What do we
Yeah.
know about an enzyme? We said an enzyme is a biological catalyst which speeds up a chemical reaction without itself being altered, which means the enzyme is never affected. Can you see that this is exactly the same? This
Yeah.
is
The enzymes
so it
pac-man.
means this is the enzyme because it's not never altered in the reaction. So straight away it means X is the enzyme. Clear? X is the enzyme. Then what fits onto the enzyme here is what we call substrates. Remember, it's a key and log substrate is this one here. W is the substrate. Clear now. So the answer becomes what? Becomes C. Even without the product missing. Clear? Makes sense? Lila?
Yes, sir.
Now look at this. Again, it's the concept of lock and key mechanism. So we are saying reactants forming what? Forming this. So now we want, let me remove this yellow thing here. I think it might be distracting all of you. Sorry about that. So now think of your lock and key. Look at this shape, look at this shape. So which can fit? This cannot fit that and that, because the reactants have to fit. So can you see that this L here? So which one is the best if you look at that? This one will fit this, but it won't fit that. So what's the answer?
E.
Fitting this and that. Remember, lock and key mechanism. Look at this. If I draw this, this is exactly this one. It's fit there. Then the small one fits there. Make sense? Lock, it's locked. Then whatever, whether it's breaking or it's forming, it doesn't really matter. Clear? So this is the lock and key mechanism. So this is not correct because it only fits this only. And there's no space for that. Clear? Lock and key mechanism. That's how enzymes work. Then this one now, people were forgetting. So the question is talking about what? Starch and salivary amylase. We said salivary amylase works in the mouth. And the pH in the mouth is 7 to 8, so it's alkaline, it's never acidic. The only region in this alimentary canal that is acidic is only the stomach. That's where you get this pH of 2. Clear. It's the only region that is acidic. So you see, most of you were saying answer. This becomes wrong here. Because why? Wrong pH. Salivary amylase works in alkaline conditions, and the human enzymes work best at 37 to 38. Clear? That making sense? It's there in the textbooks now. What is true for all enzymes? Be careful. For all enzymes. So it's not true here. But some enzymes work in acidic, some enzymes work in what? In alkaline pH. Clear? So it's wrong to say all enzymes are effective at it. Some enzymes are effective at this, some enzymes are effective at acidic pH. They are all sugars, that's very wrong. So what's the answer? D. Because all enzymes are protein in nature. Clear? All enzymes are protein in nature. Now, number 6. What could reduce the rate of production of amino acids? What could reduce the rate of production? Now it became tricky. Huh? What could produce? Now some were saying this. That's not correct. Huh? Because we said at 30 to 40 it's okay. Huh? The moment you are now above 40, you are now 40 to 50, it becomes slow. After 50, you denature the human digestive enzymes. Huh? So it's not correct here that saying raising to that is still okay. It doesn't slow. Huh? Now look, what's the pH here? 1.5. Huh? What's the pH? 1.5. They're saying the diagram shows you the effect of digestive enzyme, amino acids, protein. So we're talking of the stomach, huh? Which you said is acidic. So the moment you start raising the pH, even if I was to draw a pH, it would be like this. Sorry. The pH of that graph here, because it's best at 2 here. So it's like this. It's best between 1 and 2. Sorry. So like this. So the moment I start raising this to that, above this, I start slowing what? Slowing the production of my amino acids. Clear? The moment, remember, they work because you are moving from acidic to alkaline. You are slowly denaturing the enzyme. Which group of compounds ensures metabolic reactions take place effectively? Enzymes. Huh? Make sense? Then this were confusing some. The diagram shows you an experiment on the digestion of protein in egg augmented by protease. Okay. Now, what I need to pay attention to here is this word most quickly. Now, what do you know by high temperatures? The temperature is the same. So let's ignore the temperature. But what are we concentrating on here? Digestion of what? Protease works in acidic condition. Clear? Like your stomach. Huh? Acidic condition. So this one, acidic condition, hydrochloric acid. This one, acidic condition. This one, acidic condition. What's the problem with this? Can someone explain, Jordan? We boiled. We have destroyed what? Enzyme. So if I've destroyed the enzyme, can it break down any protein? It can't break down. Nothing happens here because we have destroyed the enzyme. Clear? The challenge with B, there is no what? No enzyme. Though the pH is correct, no enzyme to digest what? The protein. So what's the answer? C. What effect does a gradual decrease of pH, that's a good question, huh? From 13 to 1 has? So on action of what? Salivary amylase. So you need to know the graphs like what I'm saying. Salivary amylase said best between 7 and 8 like this. So if it's a pH graph, if you check your textbook, it's something like this. Huh? So it means, look at this, gradual from what? From 13 to 1, 13, 14 here. So if I'm moving from 13 to 1, what's happening here? 13 to 1, look at your graph here. 13 to 1 is what? It's increasing. Huh? 13 to 7, sorry. 13 to 7, it's increasing. Then 7 to 1 is what? It's decreasing. So what does that mean? Speeding up and slowing down. Making sense? Carry on there. An enzyme in potato cells causes oxygen, that's hydrogen peroxide, to be produced from hydrogen peroxide. Tubes of the potato were incubated with hydrogen peroxide at different temperatures. Okay, the number of oxygen bubbles released per minute were counted. So it means a fast reaction would produce more gas More gas bubbles. The results suggest that the optimum, so best pH is where you have more gas bubbles. Where do you have more gas bubbles? 96, 98, 82. There. So what's the answer straight away? That's where you have the optimum temperature, where you have more gas bubbles. Clear?
Yes.
Yes, sir.
Lactase is a human enzyme that catalyzes the breakdown of lactose in milk. At which temperature? So we said human digestive they are best at they are best at 30 as long as they are human digestive enzymes, best at 37 to 40. Right. So when is the enzyme not active? Where is it not active? There. The enzyme is not active and where? There. So what's the answer? A. Clear?
Yeah.
What happens to most enzymes above 60? They are
Natural.
they are destroyed. Now this, so when you are doing this, this is always the rate of enzyme activity. It's always the rate. The y-axis is always the rate. Anyway, the answer becomes obvious because there is no other rate of reaction here. Clear? Can be temperature or pH, doesn't matter on the other part. Now, enzyme catalysis is the same. The diagram shows the experiment to investigate the effect of changes in the pH of this reaction. The table shows you the time taken for the froth of the bubbles to reach the top. Okay, a different pH. A different pH. The time taken. So which one takes less time? This one. So if it takes less time, it means the reaction is, that's where the reaction is what? Is faster. That's all there. What is characteristics? They didn't say enzymes, or catalysts. Because in chemistry, enzymes are catalysts, but we also have transition metals as catalysts. Not only enzymes are catalysts. So if you say they are made of protein, that's not correct. It's enzymes that are made. So we have other catalysts that are metals. Clear? So they do not change. They are not changed by the reaction. They remain the same. Clear? Whether it's enzymes, whether it's metals, they are not changed by the reaction. We said they are not altered. That's true for all catalysts. Now you have this. So you now know that the enzyme is 3, everyone there. The substrate, what's fitting into the enzyme is what? Is 1. 3 and 1. Which enzyme is protease from the stomach? So optimum pH, because it's a human digestive, 37. But you see, this is wrong here. Because protease in the stomach has to be acidic. So what will be the best answer there? 40 degrees and
C.
2. Acidic condition. Number 20 then. Which statement is correct for all? So they were repeating these things. Oh, it's a bit different. They all speed up chemical what? Reactions. That's why they're called catalysts. Clear? Then you've got a 2 enzyme controlled reaction. So you can say that this one here is building up because amino acids are the smallest. This one is breaking down. So enzymes can be used to break down or to build up molecules. This is joining small amino acids together to form that. This is breaking that. Clear? So to say enzyme 1 is that's wrong. Enzyme 2 slows down production of amino, that's wrong. Can tell from the diagram. Enzymes can build up large molecules, which is correct. Huh? But we can't say they can only here. They can build up or they can break down. So the answer becomes what? C. D becomes wrong because they use the word only. Enzymes can only break down large. They can actually, you can tell from this. They can build up and they can break down. Are we together?
Yes, sir.
Yeah.
Number 22. I think only 2 people got above 95%, if I'm correct. The temperature of an enzyme control reaction is increased by 10 degrees. How does this affect the rate? So it depends, huh? Because it depends where we don't know whether the temperature is close to the optimum or is after the optimum. So it means if it's before the optimum, for example, if I have a graph for human digestion, say 37 like this, let's say 30, 37 degrees here. If the temperature was before, it will increase the reaction. If it was after, sorry, like this, it slows down the reaction. Okay, clear? So it can increase or decrease because we don't know whether it's before the best temperature, which is the optimum temperature, or it's after. It can slow down or it can increase the rate. That's all they wanted you to think along those lines. Now, in an experiment, the effect of temperature on the action of amylase is investigated. Without even reading further the question, which condition should not be kept constant? What are you investigating here? Temperature. So whatever you are investigating should never be kept constant, huh? Because that's what you want to investigate. The rest should be kept constant. Clear? Makes sense? If
Yes, sir.
if I'm investigating effect of an acid on an enzyme, I change different. If I'm investigating pH, it means I change different pH. My reaction, I expose it to different pH, but the rest should be. You can't investigate 2 things at the same time. The rest should be kept constant. So what you are investigating is what you change. Now you've got a diagram like that. And they are saying, what does this graph show you about enzymes? Enzymes work best in acidic condition, wrong, because this is acidic, this is alkaline, this is alkaline. So this is best in acidic, best in alkaline. That best in alkaline. Clear? Enzymes work best at at different pH. Enzymes work best at different pH. Clear?
Yeah.
I say, how are you? I'm good. Yeah, I've been very busy since this lockdown. Yeah, I know. Yeah, it's true. Yeah. Okay. Yeah, I think your phone is breaking or something. Yes. 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 My question is that can you also check with them if it can be done? Because I know it can be done. Everything can be done actually online. if I'm correct. Oh, okay. Yes. 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 Wow. Okay, my apologies. My apologies. Okay, are you guys there? Some plants digest insects and use nutrients. Okay, what must these plants produce to digest? So to digest, you need what? Enzymes. Huh? Clear? Then you've got a graph like that. At which pH is the activity of both enzymes the same? Where is it the same? There. At 5. Huh? Most of you got it correctly. Then number this one. Maybe some of you didn't understand the question. 4 identical mixtures of starch and amylase were kept at different temperatures. The graph shows you the time taken for starch to be completely digested at each temperature, to be completely digested. So where is it taking less time? This is temperature, sorry. So where is it taking less time? There.
B.
B takes less time. So it's most
I
effective.
didn't read the question properly.
Okay. So you're supposed to read the questions and understand them. Equal quantities of protein digesting enzymes were added to 5 cubic centimeters of protein solutions of different pH. Each tube was kept at 37. The amount of amino acid in each test tube was measured after 3 minutes. The results are shown in the table. So, okay, you see, you understand, at the same temperature, equal quantities of protein digesting enzymes were added to 5 cubic centimeters solution at different pH, so at different pH. Huh? Now, the amount of amino acids are measured in each whatever. So proteins are being digested to what? To amino acids. Huh? So it means the one with more amino acids. At which pH is it most active? Where are you having more amino acids? There.
1.
So acidic one, very acidic. Now number 29.
28 was which letter?
Did we skip 28?
No, 28
A.
was A. Okay.
Oh. Number 29. 4 test tubes were set up as shown in the diagram. 4 test tubes. In which test tube is starch digested most effectively? In which test tube is starch digested most effectively? What's the problem here? We have boiled the enzyme.
Well, I mean,
What's the problem between A and B? The temperature is too low, slow.
Yeah.
So it means this one, even if it has got the enzyme, it will be slow because the temperature. So we go to this here, good temperature. Then here we've boiled the enzyme. So what's the answer? C. Clear? Number 30. A human digestive enzyme breaks down its substrate at a fast rate. At a fast rate at 35 degrees. What could occur if the enzyme and the substrate were kept at 30 at this? You now know this. Huh? We denature it. The enzyme would stop working because it's very high temperature. Human digestive, you denature it above 50. Now, there you've got a diagram that looks like this. The diagram shows the action of amylase. What is the function of the enzyme amylase? Some of you were forgetting that we said starch. If you check your notes, everyone, plus the enzyme amylase. It's never glucose, huh? It's
Maltose.
maltose with OSE, maltose. Then it's maltose in the small intestine now plus an enzyme called maltase. That's what we said. That now becomes glucose. Clear. So what's the answer here? Breaking down, increasing the rate of that, and breaking it down to maltose. Clear. Not C. What are enzymes made of? We said enzymes, all enzymes are protein in nature. At which temperatures do most enzymes in human beings? Oh, sorry, completely denatured. I didn't read the question. So what's the answer? There, about 50. 6 test tubes were set up at different temperatures. Each contained identical solutions containing starch and amylase mixtures. The table shows you the time taken for the reaction to finish in each test tube. At which temperature does amylase work best? At which temperature does amylase work best? You use this table. Time taken to finish. So which one is taking less time to finish?
65.
You answer
65.
from, yes, we use the
Okay,
results from the
yes.
table, not from what we know from our knowledge, from the table there.
Yeah.
So what's the best answer? C. Rate of reaction, temperature. What is happening at X? The enzyme is becoming denatured, destroyed. Now this becomes a tricky question to most of you. The graph shows the effect of temperature from enzyme activity. Okay, that's fine. Which graph shows the effect of temperature on enzyme activity? Effect of temperature. You can't have the rate increasing. Remember, there is denaturing. Clear. From 20 degrees to something like 40. After that, it has to be denatured. Make sense?
Yes.
Human digestive enzymes. That's what I was looking for. Now, number 38. No, sorry, we skipped number 37. Which substance is an enzyme? A lipase. Not this. It doesn't have ASE. Lipase is an enzyme. Most enzymes end up with ASE. Yeah? If you didn't know that. Which are the characteristics of enzymes? They are protein and biological catalysts. It's there in your notes. Now let's see. This table shows you that okay, present. So you've got starch present, starch absence. Okay, starch plus water, it what? No enzyme. Clear? No enzyme. Starch plus amylase, okay, but slow. Clear? Slow. There, starch plus amylase plus 30 degrees. Okay, clear? Starch plus boiled, the boiling destroys the enzyme. So what breaks down is the enzyme, the starch. Now, what prevents proteins to change into amino acids? So what helps also? Enzymes. There have to be enzymes to break that down. Then this, it was a bonus mark.
See, there's no question here.
Yeah, I know. It was a printing.
But you still marked it wrong and you took away a point.
No, I marked out of 42. I didn't mark out of 43.
I thought on mine you
Yeah,
took
but
40.
as for me, so you removed one other mark.
I thought you marked 1 out of 43. That's
No,
why.
I did mark. Now, in enzyme, this time, that digests protein and cooked egg, okay, white that contains the protein are placed in 4 test tubes. When the egg white is digested, the mixture becomes clear. Which tube becomes clear first? So let's see what we have. So we're talking of protein digestion. Protein digestion has to be acidic, acid, acid. So alkaline, alkaline, outer. Acid, acid, okay. Temperature, this one is a good what? Good temperature. Human digestive enzymes. Clear? We are there. Then the last thing is the water bath. What's the next lesson after this?
I think it's ICT or something like that.
Environmental management, I think.
EM, we have done.
School maths.
It's called maths. Then EM. Then physics, something like that. Okay. So the apparatus shown, okay, shows that which test tube would contain most sugars after 20 what? After 20 minutes. So you see the temperature here is too low. The temperature is fine. Let's check what we have here. What's the problem with C? Is there any enzyme to digest the starch? There's no enzyme here. Here we have the enzyme. So the answer is what? Is D. Clear? Clear, clear, clear. Ella, you are there?
Yes, sir.
It's now clear now. Do you have any questions? No questions for the teacher. It's now clear? Everyone? Now, for the last 16 minutes, let's start a new topic. Then we continue the next lesson. Clear? New topic. You guys, are you there? You are very quiet.
Yes, sir.
Yes, sir.
Yes, sir.
So we have done nutrition. For those that are joining, you can read, we can send you notes if you're not there. We did a balanced diet, everything nutrition. We did food tests. Then we did digestion, the complete topic. Now we have done enzymes as a complete topic. Am I lying? I'm not lying. Huh? That's what we have done, which is a lot in a space of 2 weeks. Which is good for you. Clear? Nutrition, we covered it very well. There. Yeah, there. So we are going to the other topic that made animal nutrition. Animal nutrition. We have done digestion. Digestion is under part of animal nutrition. Then after that, we are now going to move forward to look at transport in humans. Key topic. Huh? This one might take us 2 weeks. I think 1 full week this week and next week. Transport in humans. Transport in humans. If all goes well, by the end of this first term only, would have like covered 70% of the syllabus. Transport in humans, clear? Transport in humans. So how things are transported in humans. Huh? Remember we talked about the liver, we talked about digested food going to the liver, then the liver making decisions, huh? Like what he said in terms of where the nutrients are transported and that. So now we want to look in detail in terms of transport in humans. You now know that most transport happens through the blood. Huh? I'm sure you know that. Huh? Clear? Now, so let's start with the blood. The blood. We have got 2 types of blood. What do we have 2 types of blood cells? What are they?
Red blood cells and white blood cells.
Red blood cells and white blood cells. The red blood cells are responsible for transportation of what? What's their main function? Of oxygen and carbon dioxide. Clear? This one is for defense mechanism. Huh? Clear? White blood cells are for your body defense or immune, huh? Immune system. Clear? Fighting infection. Clear? We'll talk about that in detail. Now let's start with the white red blood cells. The red blood cells. How are they adapted? So we're going to look at adaptation. How are red blood cells adapted for that function? How are red blood cells adaptation? You can write that adaptation. How are they adapted? Adaptation of red blood cells. So Tandy, how are red blood cells adapted? They have a what? A disc
shape.
Disc shaped large surface area. Disc shaped large surface area, so that they can absorb more. So to absorb more oxygen. Huh? To absorb more oxygen. Clear? Red blood cells are disc shaped. What else? Besides being just disc shaped, do they have a nucleus? No, no nucleus. No nucleus. What's the advantage of no nucleus? More space. Huh? More space. Blood. No? More space for the blood and your oxygen. Now, what else? They've got hemoglobin, which is a red pigment made from iron. Hemoglobin. So, what's the function of hemoglobin? What's the function? Who can tell me the function of hemoglobin?
For the red pigment on the...
So hemoglobin combines with oxygen to form oxyhemoglobin. In biology, that's what you call oxygenated blood. Clear? Oxygenated blood is oxyhemoglobin. Clear? It also combines with carbon dioxide to form carboxyhemoglobin. In biology, we call that deoxygenated blood. Clear? But deoxygenated blood is actually carboxyhemoglobin. Clear? They are making sense?
Wait, how do you spell carbon, whatever?
Carboxyl. Sorry, XY. Then a hemoglobin.
Okay.
Clear?
Yeah.
Makes sense now. So maybe what we need to know, or just to write that, is that oxygen diffuses from alveoli, which is your lungs, from alveoli in the lungs. Oxygen diffuses from alveoli in the lungs into the red blood cells, and combines with the hemoglobin, forming an unstable compound called oxyhemoglobin. Combines with the hemoglobin, forming an unstable compound called oxyhemoglobin. Clear? When the blood reaches the body cells, the oxyhemoglobin is easily split to hemoglobin and oxygen again. Clear?
No.
When
Please repeat, sir.
when the blood
Yeah, I got lost.
reaches the body cells, when the blood reaches the body cells, the oxyhemoglobin is easily split into oxygen and hemoglobin again. Is easily split into oxygen and hemoglobin again. Clear? And hemoglobin again, so that the oxygen is used for respiration. So that the oxygen can be used by respiration. Because that's its
Yes,
function.
sir. Can you please repeat?
Which part? Which part exactly? When the body reaches the body cells, the oxyhemoglobin is easily split. The oxyhemoglobin is easily split into oxygen and the hemoglobin again. Into oxygen and hemoglobin again. The oxygen is used for respiration. That's what I think. Clear?
Yeah.
You're going somewhere? So we are done with that. Just hold on there.
Okay. This is the
Hi,
best
boss.
information.
Hi. Now let's look at white blood cells. White blood cells. White blood cells. Yasin, are you there? He's not there. White blood cells. What are the two types of white blood cells?
Don't know.
What are they? Phagocytes and lymphocytes.
Lymphocytes.
Phagocytes. Phagocytosis. Phagocytes and lymphocytes. Four sites. So these are the two types. So you need to know them in terms of shapes, to be able to identify them that this looks like a phagocyte. I'm not good at diagrams, but I think it's something like this. Then the other one will be something like this. Clear? That is there in your book, sir.
Yeah,
So
can I just
this,
find in my document
yes,
board?
you can always draw from your book. So this is called a process called phagocytosis. Phagocytosis. We need to explain it. Phagocytosis. Phagocytosis. So phagocytosis, we're now to explain them in stages. What happens is that the phagocyte engulfs step one, under phagocytosis, step one. The phagocyte engulfs bacteria, clear? The phagocytes engulfs bacteria. They engulf bacteria, clear?
Yes, sir.
By taking them into the cells. So that's engulfing. Number two, they secrete enzymes to digest the bacteria. That's all you need to do. Number two, they secrete enzymes to digest
Come again.
bacteria. So the first thing is that this phagocyte here, this space that you're seeing, it will engulf and close here. It engulfs the bacteria. When it engulfs the bacteria, it now secretes enzymes. Because the bacteria is being engulfed, it can't run away the pathogen. It now secretes enzymes to digest the pathogen. Hi, in dream lesson. Hi, in dream lesson. It just text. So is that clear, everyone there? Or I
Yes,
need
sir.
to explain again? It
Ah,
engulfs
it's clear.
the pathogen,
Yeah.
then it secretes enzymes, they digest. That's how they kill infection. Huh? That process is called phagocytosis. Clear? Phagocytosis. Now let's look at lymphocytosis. 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 Lymphocytosis, clear? Now, under lymphocytosis is you can write that in summary, the lymphocytes kill bacteria by secreting antibodies,
So can you please come again?
These lymphocytes kill bacteria by secreting antibodies and antitoxins, which kill bacteria. They kill bacteria directly. Huh? They kill bacteria or pathogens directly. These ones actually kill directly because remember, every pathogen has got what you call antigens, shapes. Huh? So these antibodies will create a shape that is complementary to this, that will lock this. It's more like a lock and key.
Like the lock and key method.
Yeah, but we'll talk about that under immune system, not now. Clear?
Yeah.
Makes sense? So the blood, we now know that the blood is made of red blood cells or white blood cells, huh? But that's not the only thing. The blood has got also what are called platelets. Huh? Oh, we don't have time for that. I'm just looking at time. Platelets.
All right, platelet.
Platelets, which look like tiny. These are tiny fragments. Let's quickly write that. Tiny fragments which prevent bleeding when the skin is cut. We need to know how that happens. These are tiny fragments which prevent bleeding when the skin is cut. Clear? When the skin is cut. Clear? Now I'll explain the biology, then we end of what happens. Huh? Clear? When the skin is cut, the platelets, causes fibrinogen, which is a protein. It causes soluble fibrinogen.
Wait, which
When the
causes?
skin is cut, the platelets causes soluble fibrinogen.
How'd you spell that?
Soluble fibrinogen. To be converted into insoluble fibrin. To be converted into insoluble fibrin. Clear? Insoluble fibrin. Are we together?
Yes, sir.
The next point is that the fibrin forms long fibers. The fibrin forms long fibers that clot together, blocking the cut. That's what you call blood clotting.
Sorry.
The fibrin forms long fibers, then we are done, long fibers that close together, blocking the cut. We are done here. You can read in that textbook, bio notes is also explained. We'll see you
Close together, sorry.
to stop the cut, to block the cut.
Okay.
We are done. We need to know that process here of blood clotting. We are done for today here.